Um, Good day, everyone. For our exercise number 3, the topic is histopathologic techniques. So, what is the importance of histopathology? So, in histopath, we use histopathology in order to examine the microscopic lesions, specifically lesions from tissues caused by infectious or non-infectious diseases. Or in another meaning is we need to know the effect brought by the organism to the tissues of the animal. Histopath is also used for diagnosing tumors or identifying tumors and the nature of it, whether it is cancerous or not. So in histopath, this involves more on the tissues. So study of tissues can be obtained from different means. Now, so we can obtain in clinical like fine needle biopsy, or you can have an excisional biopsy, and by taking the excisional biopsy, you have to get the normal and the abnormal tissue. So necropsy and autopsy, how is it how is it different? So some may be confused why there is autopsy and why there is necropsy. So when you say necropsy, this is commonly used in animals. So necro means death, and autopsy commonly used by human medicines or humans. So auto means self. So just like autoimmune antibodies. So in histopathology, first is after you have to get a tissue or sample from the lesions from the animal that you examine, from the gross lesions. So first is you have to get the gross lesion and then remove it. And then after being removed from an animal, a tissue or organ is cut into pieces, and then these pieces are placed into a fixative agents like such as buffered formalin or 10% buffered formalin, which ideally preserves for the normal morphology and facilitates further processing. So after fixation, the specimen is dehydrated, and by transferring it to a series of alcohols of increasing concentrations until 100% alcohol. Next is it is placed into a substance such as silane or silane substitute, which is miscible, meaning the property of two substances can be mixed. So which is miscible for both 100% alcohol and paraffin. So this intermediate step is called the clearing, which is essential before infiltrating the dehydrated tissue with paraffin because alcohol and paraffin do not mix. So during infiltration, melted paraffin completely replaces the silane. So this procedure is done in an oven at a temperature just above the melting point of the paraffin. So when infiltration is already complete, the specimen is transferred to an embedding mold of fresh paraffin, which is allowed to harden. Then the mold is removed and the excess paraffin is trimmed away. Then the block of the paraffin is then secured to the microtome and oriented appropriately with respect to the knife, With each revolution of the microtome handle, the specimen moves through the blade and a section of the desired thickness is produced. Each successive sections adhere to the preceding one, forming a continuous ribbon. So this is also called as section or ribboning section. Subsequently, one or more sections are carefully separated from the ribbon, and then you transfer it to the surface warm water in water bath. So the purpose here is to soften the paraffin and flattens the section, or can have a good orientation of the section. And also the very most important is to eliminate the wrinkles of that section. And then the flattened section is fluted into a slide, which is then placed on a warming table. As the preparation dries, the section adheres to the surface of the slide. Next, the paraffin is removed with xylene or another appropriate solvent, and then the specimen is rehydrated. And after this, it is then stained and then dehydrated and cleared to make it transparent with xylene. And then after that, cover it with a resinous mounting medium and top with a cover slip. So here, this is figure 1.2, B, A, B, and C, and figure 1.3, which shows a variety of three-dimensional structures and how they would appear if cut at different levels or at different view. So examine this carefully, and they will help you to understand situations you will encounter on actual slides. And so here, cells are three-dimensional objects differing in size and shape. For example, some are long and thin, and some are cuboidal, and others are ovoid. So they may have a random or specific arrangement within an organ. So how they appear depends on their shape as well as how they were cut. Imagine how the spindle shape and the tall columnar cells, which is shown in this figure in various planes. You note also the nucleus may or may not be included in a particular cut through a cell. So the histologists examine a multicellular structures having a wide variety of shapes. Some are hollow, some branch repeatedly, and some open onto surfaces, and etc. So I'd like you to see the video presentation of how the histopath procedure processing take place. I want you to observe the video, and then after this, I will discuss on the details of this exercise. Thank you. So in this exercise number 3, which is entitled histopathologic techniques, no, the objective tips for this at the end of the exercise, the students are expected to know and understand the importance of histopathologic techniques and their purposes. And then second is to illustrate and discuss the following histopathological procedures. So such as, which I've mentioned earlier, fresh tissue examination, fixation, decalcification, dehydration, clearing, impregnation, trimming of sections, cutting of sections, and then staining it to a specific stain, uh, which are hematoxylin and um, eosin stain. So number two, discuss the importance and purposes of histopathology and its techniques, and then give at least 10 situations and their applications in the field of veterinary medicine. Number three is to illustrate and discuss the following histopathological procedures. No? The fresh tissue examination, fixation, decalcification, dehydration, clearing, impregnation, embedding, trimming of sections, cutting of sections, and then staining, uh, specifically the hematoxylin and eosin stain. So please include the purpose of each procedure in the discussion.